So really quickly before we get started, our seminar speaker tomorrow in the biology department, noon in, I think it's room 101 in SRTC, is Harmeet Malik from the Fred Hutchinson Cancer Research Center that's up in Seattle. The title here, unfortunately, doesn't say that it's all about viruses, which hopefully it should do. Genetic conflicts, the usual suspects, and beyond. He's extremely well known for studies of what he termed paleovirology. So basically going back in time by using sequences to try and reconstruct what kinds of interactions hosts and viruses had in the past. And particularly what he's been looking at are the host responses to viruses, in particular, again, the retroviruses. So what we'll be talking about today and the ApoBec interactions with virus factors and how there's sort of been coevolution between the two, or an arms race, as some people call it, between host defenses and virus attack mechanisms. So that process is what he's going to be talking about tomorrow. Yes?
What time is that?
Well, the time tomorrow is noon over in SRTC. I understand other people may have other things that are happening then, but I've heard him speak a couple times. He's also been on This Week in Virology, TWIV. Is generally extremely interesting, great talk, really cool stuff that he's doing. So that being said, let's move on to our PowerPoint for today. Again, it's retroviruses. If you'd like to learn more, I have this small book here on RNA tumor viruses, which is basically the only reason that people were interested in retroviruses to start with, of course, until HIV came along. But before that, we need to talk about influenza, or you get to talk about influenza, because we have our clicker questions. Influenza nucleocapsids are transported to the nucleus because they're encapsulated at the nuclear membrane. That's where the caps are. They require cellular DNA polymerase. They require cellular RNA polymerase. They require tRNAs. Now, the key is to sit far enough away from somebody that you're not going to get infected by whatever they have, but so close enough that you can tell them, talk about the answers to the clicker questions. 3, 2, 1, lift off. So, yes, it's where the caps are. Now, that is true to some extent. What's interesting is that none of you asked me questions about, well, there are messenger RNAs in the cytoplasm too, right? And those all have caps as well. So, potentially not as concentrated. If you think about, so, why, again, go to the nucleus? They're not as concentrated. That you could argue about. If you think about the eukaryotic cell, often the nucleus is just about as big as the cytoplasm. So that's one thing. But what's much more important about the mechanisms that influenza is using? Exactly. It inhibits tailing. And that's all happening in the nucleus. And that means you're accumulating the caps, particularly in the nucleus. And the reason I mention this is one of the viruses we didn't talk about, the Bunya viruses, which I was going to talk about after filoviruses, but got too carried away with Ebola and those kinds of things. Those actually steal caps in the cytoplasm. So there are viruses which will get caps in the cytoplasm, as well as influenza, which is picking them up in the nucleus. Encapsulated the nuclear membrane, there are viruses which are, but where are influenza encapsulated? The plasma membrane, exactly. Do they require cellular DNA polymerases? They're RNA viruses, so they certainly don't need cellular DNA polymerases. Whether they require a cellular RNA polymerase, one could probably argue about this. I realized just a second ago that it's the cellular RNA polymerase which is making the messenger RNAs that then get the caps put on them. I think B is a better answer than D, but again, one could argue about that. They require tRNAs. Well, eventually they require tRNAs, yes, but you also have, of course, all the tRNAs in the cytoplasm as opposed to the nucleus. As we'll see today, talking about the retroviruses, these require tRNAs for their replication, as well as making all of the proteins that you happen to need. So, next question. How many different proteins are in influenza A virus virions? 1, 2, 4, 6, or 9? And no, you don't get extra credit if you name all of them.
Why are pharmaceutical companies taking vaccine if the strain isn't very common? Just to like marketing or?
We can talk more about that later. That's a very good question. 10, 5, 4, 3, 2, 1. Beep. E, yay. This gets back to a question somebody asked me right before class, and that is, when you're studying for the exam, should you be grabbing the textbook and reading through page by page, word by word, highlighting absolutely everything? No. You should be looking at slides from my lectures, because that's what I do when I put the questions together. So this, again, looks great. Uh-oh. Did I actually choose the right one? That might help for my scoring processes. Okay, our last clicker question for today. Seasonal inactivated influenza virus vaccine in the Northern Hemisphere is made from bird flu, 3 or 4 different human influenza strains, an influenza strain with 3 or 4 different HA and NA proteins, recombinant HA proteins, or isolates from the Southern Hemisphere. $15, 10, 5, 4, 3, 2. Okay, we've got some differences of opinion on this one. Trick question. Oh, they're always trick questions, right? Stedman's exams are always trick questions. So a couple of important aspects about this question. One is the inactivated influenza virus. If I'd said attenuated, which one would that be? So it would be C in that case, because they're reassortments where they just put in HA and NA on the background of the cold-sensitive virion. But it says inactivated. Northern hemisphere, well, they're based on what's used, based on what has been circulating in the southern hemisphere, but it's not necessarily made from isolates from the southern hemisphere. And so a lot of, if you look at the current vaccine, then it's partly northern hemisphere, partly southern hemisphere. And so it's not being made from one versus another. 3 or 4, why do I have to put 3 or 4 in here? Well, no, because we've got a trivalent and a tetravalent vaccine that are currently on the market. So one of the B strains or 2 of the B strains. And somebody asked a question up here about the B strains, and they cause much less disease. Why do we bother to have them in the vaccine in any given year? The data that I showed, particularly from Oregon, if I remember correctly, there's a bump right about now of people with influenza B, and those are from hospitalizations when you look at those particular diagnoses. So yes, it causes less disease, but it's still causing some hospitalizations. So that is clearly an issue there. Bird flu would be if we're talking about pandemic influenza, which we did not talk about. And by the way, if you looking at the PowerPoints which I posted, I've got a whole bunch of stuff on pandemic influenza. Fascinating, interesting, important, not going to be on the exam. So ignore it for the time being and go back and look at it in your copious free time thereafter. So yes, our answer is B, because it's these different human influenza strains which are used to make inactivated influenza. So those people who need to run away, adios, amigos. But otherwise, in terms of orthomyxoviruses, again, this is basically influenza. Everything that we know about influenza and anything that we know about these orthomyxoviruses really comes from this study. And they're these interesting multipartite negative strand genomes. And so this is the big difference between these viruses and really any of the other ones we're going to talk about at all this term, is this segmented negative strand RNA genome. And so again, the fact that you've got these segments allows recombination to take place, and it's just shifting of the different chromosomes around, or of these different genome segments. So that's what's being used in the live attenuated vaccine, and also where we have problems like the swine flu from 2009, which is just 2 of those segments which are clearly a pig-related virus, one which is from an avian influenza, and then the other 5 which are much more of a human influenza variety. So there are questions, more questions about the orthomyxoviruses. The other big aspect here, of course, is that many more people die from seasonal influenza than probably almost any other virus, with the exception now of HIV. Okay, so retroviruses. These are, again, switching from our negative strand back to the positive strand, but these are completely confused viruses because they make DNA as well as their RNA. So here, with a classic retrovirus, these are going to be packaging your positive strand RNA, But as we talked about way back at the beginning, when you think about retroviruses, or at least viruses which replicate through an RNA intermediate, they can package DNA, they can package RNA, they can even package, in some cases, combinations of the two. Most of what's known about retroviruses, again, before the advent of HIV, comes from study of RNA tumor viruses. Hopefully, all of you know that cancer is a genetic disease. What's cellular genetic material? DNA. But these are RNA viruses. So how can RNA viruses be causing tumors? And this was a big, huge question for long periods of time. People knew about these RNA viruses which were causing tumors. Peyton Rouse in the early 1900s had discovered these things. But they were RNA viruses, and cancer was a genetic disease, and genetic disease is all about DNA. And so it was only in the late 1970s that people discovered reverse transcriptase, which was taking an RNA copy and then making it into DNA. And so it was really in the late 1970s that people finally discovered that the reason these were RNA tumor viruses is that they actually also had a DNA part of their replication process. Then, of course, HIV appeared coincidentally rather soon thereafter in the 1980s. And most of the work that's been done on retroviruses since then has been done on HIV. There are a whole bunch of key concepts about retroviruses, and we could easily do a whole course on retroviruses. I mentioned the tRNA primer. This is the cellular component which is absolutely required for replication of all retroviruses, be they HIV or any of these other retroviruses. And so, as true with pretty much any virus that has a DNA intermediate, and even a lot of the RNA viruses which use polymerases, which are very similar to DNA polymerases, you need a primer. And so the big problem with getting replication is getting a primer. There's a whole bunch of tRNA around in the cell, and retroviruses use these tRNAs as primers. They also use frameshifting. We've already talked about frameshifting in terms of looking at some of these negative strand RNA viruses, the paramyxos, the filoviruses, etc. So it's pretty similar in terms of how that's concerned. What's quite different in terms of these viruses is they use splicing on a much greater extent. We talked about splicing in terms of influenza viruses last time, but here splicing is absolutely critical for the replication of these viruses and making the virions. Reverse transcription, of course, this is going from your DNA to RNA. What does that? It's the reverse transcriptase. Why do you go through this whole process of making DNA? Well, it's because the virus genome gets inserted into the host genome, so that integration process. So it's not just going through reverse transcription, which is important for these retroviruses. It's also the integration of that genetic material now into the genome, which of course is what can cause these tumor viruses to take place. To get that integration, but also part of the replication mechanism, you have these things called LTRs, or long terminal repeat structures. So at either end of your genome, you have these repeated sequences which vary from retrovirus to retrovirus, but they're between a couple of hundred and 1,000 or so nucleotides at either end, which are identical to each other. We'll see how that happens due to the replication process. And that's absolutely critical in terms of how you make these things. It's also important for getting integration to take place, to have these identical sequences at either end of your replicated genome. Template switching is also absolutely critical in terms of retrovirus replication. And then just the nomenclature here, GAG, POL, ENV. Unfortunately, that's what the nomenclature is for all of these viruses. GAG are the group-specific antigens, because they were mostly found, again, in tumor viruses. Which group is it? You know, what are these being produced? POL, of course, is the polymerase, but the POL genes also include the integrase gene. And then the ENV are just the envelope proteins, which are involved there. So again, we'll start out talking a little bit about origin and diseases. We'll talk about HIV towards the end of the lecture today. Look a little bit at the structure and entry, but the big important aspect of how these guys make more virions has to do with their replication process. A little bit about transcription, a little bit about assembly and export. These are some of the retrovirus virions. These are all examples of HIV, clearly enveloped and have a nucleocapsid, which is present on the inside. What kinds of retroviruses do you have? Again, most of the work that was done before the 1980s was on various different sarcoma, leukemia, tumor viruses, etc. Then, of course, we have the immunodeficiency viruses. These are also known as the lentiviruses or the slow viruses. They take a long time to replicate. And we'll see why that is a little bit later on. They also take a very long time to cause disease, whereas most of these tumor viruses are much more rapid in terms of causing disease. So both slow replication, but also giving you this very slow disease phenotype. A classic retrovirus looks like this, and the big difference here is really in terms of the structure of the nucleocapsid. Most of those RNA tumor viruses have icosahedral nucleocapsid structures inside an envelope. Inside that nucleocapsid, you have 2 copies, and you always have 2 copies of the genomic RNA, and we'll see why that is a little bit later on, but it's mostly because you need multiple templates in order to undergo your replication. You also have cellular tRNAs, these little cross structures which are associated with the genome, and we'll look at that in a little bit more detail. Even though these are positive strand RNA viruses and in theory could be used directly for translation, because they go through this reverse transcriptase step, they also have to have enzymes that come with them. And those enzymes are basically the reverse transcriptase and then the other enzymes, the integrase, etc. So all of the enzymes are packaged together in these virions. So they're a lot more like the negative strand RNA viruses in terms of what's being packaged in the virion as opposed to a classic positive strand RNA virus. Envelopes are pretty standard. They've got the matrix protein. They have a surface and transmembrane protein. People call them SU and TM. But they're basically receptor binding proteins and also contain fusion proteins. Again, pretty standard in terms of how these guys are put together. They're a little bit different because they're made as polyproteins as opposed to, certainly for the influenza viruses, where they're made from different segments. If you look at the genome, they're all set up in pretty similar ways. Again, this is a very general retrovirus structure. You have the GAG proteins here towards the 5' end, Pol proteins in the middle, and then the envelope proteins down at the 3' end of your genome. What's packaged inside the virion has a cap and a tail. Those are all from cellular, normal RNA polymerase, capping enzymes, tailing enzymes, etc. The GAG proteins contain most of the structural proteins, matrix, nucleocapsid, and capsid. Nucleocapsid is what's binding to the RNA. The capsid protein is making the capsid structure underneath the membrane. Then you have all of your sort of classic non-structural proteins, the Pol genes, which include reverse transcriptase and integrase, but also include protease. And that's because this, very similar to the other positive strand RNA viruses that we've talked about, these are all made as polyproteins. And so you need a viral protease to chop these things apart. And in fact, currently one of the best anti-HIV drugs is an inhibitor of this particular protease. Down at the 3' end, you've got the other structural proteins. These are your membrane proteins, the surface protein, and the transmembrane protein. So those are the protein coding genes that you have in these retrovirus genomes. Mentioned before, you have these repeated sequences at either end of the genome. R, again, incredibly creative nomenclature, the repeated sequence that you have at the extreme 5' end and the extreme 3' end of the genome. Then you have some unique sequences, U for unique at the 5' end, and unique sequences at the 3' end. And then a primer binding site right before your main splice site. This is your 5' splice site. The 3' splice site you can see over there. That gives you your envelope proteins, so SU and TM. So the main splice site will happen here, splice over to that end, and give you your envelope proteins. The only other thing to mention on here is this funky Greek letter, the psi sequence. This is the packaging sequence. And so this is where your nucleocapsid protein binds to your genomic RNA in order to get packaging to take place. One thing to notice here immediately is it is 3' to the 5' splice site. So when you have splicing, you're not going to have this RNA, so you're not going to be packaging that spliced RNA. So the packaging has to happen in a non-spliced RNA. If you look at the replication process, and this is the overall replication process, we'll talk about the whole, reverse transcription up at the top here. This, by the way, is from the Flint textbook, where they've got, I think, really nice overviews of each of these different viruses and their replication patterns. You have binding to receptors on the outside of the cell, fusion that either happens in, from 1 is the binding and 2 is fusion. Fusion can happen either at the plasma membrane or in the endosomes, change in pH that happens. So it's either binding to the receptor that causes the conformational change or a pH change. We saw that way back when we looked at some of the anti-HIV drugs that were involved in that change in process. Once you have a release of the nucleocapsid, it turns out in the nucleocapsid is where you start to have reverse transcription taking place. And so the nucleocapsid itself is permeable enough to nucleotides that it can actually do all of its reverse transcription out in the cytoplasm and basically in these particles. That eventually gets broken down. You have your double-stranded DNA that then gets transported into the nucleus. It can either be transported directly into the nucleus. That's what happens with HIV. Most of the RNA tumor viruses, turns out they wait until cell division takes place, and then they get inside the nucleus. Because this double-stranded DNA is pretty big. And so getting it inside the nucleus is a bit of a challenge because usually can't get through the nuclear pore complex. So once it's inside, number 5 here gets integrated into the genome. Then in the genome you have cellular RNA polymerase, which will make either the full-length genome, which gives you GAG and POL, or the spliced form, which is going to give you TM and SU. These guys get transported out to the plasma membrane through normal processes, membrane proteins. The GAG protein you make a whole bunch more of than you do of GAG and POL, having to do with the frameshifting that takes place in the middle of the genome. So these are your structural proteins. These are the enzymes. These all still, however, have to be packaged into the virion, because if you don't have these enzymes here, you're not going to be able to go through that whole process at the beginning. This then binds to that psi sequence. You get packaging. The whole virus is released. The viral protease, curiously enough, most of its activity happens outside the cell. So the viral protease, the activity of that viral protease happens once you already have the virion that's taken place. And probably the reason for that is most of the enzymatic activity that you need for doing this whole replication process at the beginning, that's already in here. And you don't need those enzymes until you have another infection that actually takes place. Yeah?
This is slightly going into immunology or something.
Mm-hmm.
But with HIV, it infects the helper T cells, correct?
Yes?
So do those divide themselves? Don't they come from like a venison type predecessor cell?
Yeah, that's, as you say, it's definitely an immunology kind of question. We may get there towards the end, but let's talk about general retroviruses first. But yeah, and it turns out for HIV, there's a very specific import mechanism that gets into the nucleus in the absence of cell division.
Yeah, basically what I'm asking is that why they don't nucleus directly, since they don't undergo cell division?
Presumably that's why, yeah. So, but again, that's, okay, don't bother about that in terms of the midterm. That's an immunology question. So, but yeah, so again, and then you end up with the protease happening out here at the outside. But the real critical aspect is what's going on up there in 2 and 3. How do you go from 2 copies of a positive strand RNA to double-stranded DNA? This is the process here. Rather convoluted, but it works. And true with all of evolution. As long as it works, you can use it. So right up at the top here, start out with the genome, that we bring into the cell in the first place. So you've got a tRNA bound to the primer binding site, which is way down at the 5' end of the genome, which is really kind of a bizarre place to have your primer, right? Because you're supposed to replicate the whole thing. Well, if it's at the 5' end, all polymerases go from 5' to 3'. So it's going to replicate from there. So the brown, which is shown up here, is the first piece of replication that takes place. So you start out from your primer, and you make some DNA, which is what the brown stands for, out to the end of the genome. It's a really short piece, actually. And again, it depends on your retrovirus, but on the order of a couple of hundred nucleotides. Then, one of the critical aspects of the reverse transcriptase is it not only has RNA-dependent DNA polymerase activity, but it also has RNase H activity. And RNase H is an RNase which cleaves hybrids between DNA and RNA. So it chops up all of the RNA template there where you just made a DNA copy of it. Well, now you have a complementary sequence here to that repeated sequence, which is over at the other end of your genome, or the other end of the other genome, which has come inside the cell as well. So you remember there are always these 2 copies of the genome which come in. So this may be going and binding to the other copy of the genome, or it may be just circularizing on that same template that it started with. So now you have a RNA here, which is complemented to the DNA, which you made at the very beginning. And this is great because now we're really way down here at the 3' end of your genome. Now you can replicate your way all the way through to the opposite end of the genome. So now you end up with a full genome copy. But what you've done is you've taken the sequence which you had at the 5' end of your genome and shifted it over to the 3' end of the genome. So you end up with this sequence, unique 3', repeat sequence, unique 5'. So now you've got a single-stranded DNA, which is appropriate for your whole genome. But you still need double-stranded DNA if you're going to integrate it into the host genome. So now you have, again, degradation up at step 5 there of almost all of your template, except for one little segment. This is on polypurine tract, which is resistant to RNase H digestion, and resistant enough that it provides a 3' OH that the reverse transcriptase can bind to and extend out through the sequence in your primer, which is what bound to the primer binding site way back over here at the 5' end of your genome. So now you've got a DNA copy of that primer binding site. RNase H slowly cleaves off that PPT and the rest of your tRNA. So now you've got a short piece at the opposite end, which has your primer binding site, which of course now is complementary to the primer binding site that you had at the other end of the genome. So you either loop around, you go back to the other template, and you replicate all the way through the end here. But now you've got a 3' end right here and a template. So that replicates out the other direction. So in this process, you've gone from single-stranded RNA to double-stranded DNA, and you've also made a copy of the repeat from the 3' end and popped it at the 5' end, repeated this 5' sequence, and popped it down at the 3' end of the genome. So that's your long terminal repeat sequence. So the LTR sequences that you have down here at the end. Another thing, I added my notes down here at the bottom. Reverse transcriptase, like pretty much all of the viral RNA-dependent RNA polymerases, is very low fidelity. So it puts in a whole bunch of mistakes. And this is one of the classic issues with trying to deal with anti-HIV therapy. There are always lots and lots of point mutations that happen during this replication process. And of course, it's now made 2 copies of the genome. So you've got 2 opportunities to make mistakes and usually not get corrected. And so what you end up with is, instead of one genome that has been repeated exactly multiple different times, you end up with a whole population of genomes, each of which has a couple of different point mutations in it, single nucleotide changes. And this is what's called a quasi-species. And it's true for retroviruses, but it's also true for pretty much any other RNA virus, because anything that doesn't have nice proofreading activity, you're going to have a number of single nucleotide changes in any population. Anytime you purify virus from a culture, you're going to have mostly one sequence, but lots of variation in that individual sequence. So it's this whole idea of a quasi-species that is what you have here. The reverse transcriptase itself is really the central enzyme in doing this process. A whole bunch of copies of it in the virion. It works in just virions itself. And so I mentioned before that this reverse transcriptase seems to take place in capsids. So once you have membrane fusion that takes place, reverse transcriptase can go from there. Originally, it was found in purifications of virions. And so people showed the activity of the reverse transcriptase in these purified virions. It has RNA-dependent DNA polymerase activity. This is what most people think of when they think about reverse transcriptase. But it's also being used to make that second strand of DNA. So it's a DNA-dependent RNA polymerase. At the same time, DNA-dependent DNA polymerase as well as an RNA-dependent DNA polymerase. It doesn't make RNA, sorry. Also as a DNA helicase, pulls the 2 strands of DNA apart. It also works as a DNA-RNA helicase because that's what it needs to do to replicate. And it has this RNase H domain as part of the protein. So 4 activities in one enzyme, which is pretty amazing. That we've got all of these different activities. But basically, we've already talked about the DNA-dependent DNA polymerases and RNA-dependent RNA polymerases as having this very similar structure. And it turns out that the reverse transcriptase also has an extremely similar structure to that. And that's what's shown here. Unfortunately, rather hard to see the structure here. This is the structure that was determined together with an RNA-DNA hybrid. So this is the reverse transcriptase up here at the top. It's actually a lot easier to see in the cartoon form here down at the bottom. This is the structure of the reverse transcriptase from HIV-1 in the absence of the RNase H domain, which is up at the top on the right here. But the point here is really that the poliovirus polymerase, which of course is an RNA-dependent RNA polymerase, is extremely similar to this structure, as is the structure of DNA-dependent DNA polymerases. So this sort of hand structure with fingers, thumb, and the palm, and the active site is in the palm of your polymerases. This is a very standard structure and probably the original structure in terms of any of these polymerases that was used. So once you've made your double-stranded DNA, now you need to insert it into the host genome. That happens through the integrase protein. Integration is said to be random. This is, in fact, one of the problems with gene therapy. A lot of people have used lentiviruses for gene therapy purposes. Also, if you think about RNA tumor viruses, lots of times they're inserted into active parts of DNA. So if you think about, usually people draw the DNA as, nice, again, double-stranded black line here. Of course, it's not what it looks like. It's compacted into multiple layers of chromatin. And so probably integration takes place in areas of chromatin where the DNA is relatively exposed. And so actively transcribed parts of DNA, which is why there are potential problems with RNA tumor viruses, et cetera. So the integration process, however, takes place through the binding of the viral integrase protein to those repeated sequences at either end, the LTRs. It chews off a couple of bases at either end of the genome and then binds to a sequence, and this should be any sort of random sequence that you have here in the genome. You get phosphodiester bond linkage. That's what the integrase does. Then that integrates the genome into the host genome. These extra bases, which are not base pairing, that were cleaved off in order to give the integrase a 3'OH to hook up the phosphates to. These then are removed by cellular repair machinery, and you end up making copies of that sequence, which was on the outside of your genome to start with. Whenever you bind, this is just like what happens with transposons. When they hop into the genome, it cuts in an offset position, but you link or you put in your transposon in there as basically blunt-ended. So you end up with these gaps at either end of your inserted virus or transposon. Those get filled in by cellular repair machinery, and you end up with direct repeat sequences on opposite ends of your inserted genome. These are now direct repeat sequences that came from the host genome. You also, of course, have these LTRs, which are direct repeat sequences which are present in the virus genome as well. So two different kinds of direct repeats. There's an OK video for integration here on YouTube. We're not going to go to it right now. I've been looking for a nice animation that goes through all of the whole reverse transcription process. I've yet to find one that does a really nice job of that. But this one does a nice job. So it talks about reverse transcription in here. I don't like that because it doesn't have the appropriate level of detail, I think. But here, this is pretty good for the integration process at the end. So now you're integrated. How do you make more virus? Well, happens through cellular transcription. Why do you bother to, again, totally over anthropomorphizing here, integrate in the genome? Well, A, you get maintained in a vertical fashion, so a lot like lambda and the lysogens, but also you can take advantage of the cellular transcriptional machinery. Turns out that these LTRs are really, really, really good promoters. So they bind very well to general transcription factors and then get transcription to take place, and that transcription happens right at the beginning of this repeated sequence. So you have transcription, two different kinds of transcripts. You either get full-length RNA, which has your GAG and POL and also ENV proteins in it, or you can have splicing that takes place, which generates your envelope messenger RNA. Caps and tails, they're all cellular processes. Again, these are made just like the other positive strand RNA viruses as polyproteins that then get chopped up. Polyproteins here, GAG and POL, each of those are individual proteins that then get chopped up by the viral protease, but you also have this frame shifting that takes place between the GAG and POL genes. So this happens about 10% of the time for most retroviruses. 90% of the time, you stop right here at the end of those GAG proteins. So 90% of the proteins that are made through translation are made then as this 5' end of that messenger RNA. 10% of the time, you have a frame shift that takes place due to some kind of secondary structure. That secondary structure could be a hairpin loop structure, as is shown here. You also have pseudoknots, as we talked about before, in terms of getting frame shifting to take place. And basically, it's confusing the ribosome, so it bounces back a couple of nucleotides. Usually that's because you have other possibilities for base pairing to take place. So in this particular case, phenylalanine, UUU, could be base pairing in two different places here. Ribosome, you can think of it running into this structure, then bouncing back a little bit and keeping going, again, about 10% of the time, and falling off, giving you translational termination 90% of the time. You get, again, these polyproteins that are formed. They get cleaved. The envelope protein is cleaved into TM and SU. That's what's shown down here at the bottom. The GAG protein to matrix nucleocapsid and capsid. And then the Pol protein, protease, the reverse transcriptase, and integrase proteins. These guys get targeted to the plasma membrane through covalent modification. We've talked about this before. This is a fatty acid residue that gets added to the GAG proteins, particularly the matrix protein right here, which then will target, and it turns out it targets the polyprotein to the membrane. And it's at the membrane that then you get these extra cleavages that takes place, or even in the capsid itself. The nucleocapsid protein binds to this psi sequence, which is, again, only in your full-length RNAs, because spliced out it's not there. And again, the protease is active only at the very end of the process. So now I'd like to talk about a couple of more specific retroviruses, particularly the tumor viruses. And there are basically two flavors of tumor viruses. This is just the one here, which is probably the most famous. These are the viruses which have included oncogenes into their virions. So what seems to have happened in this case is there's been integration of one of these retroviruses into a genome next to an oncogene. In this case, it's the Sarc oncogene. Here's an ERB oncogene. So these are genes that are, when misexpressed, lead to overproliferation of cells. So it's probably an integration that took place there. Then you had transcription of this whole piece of RNA. That then got packaged, was brought back in through reverse transcription, ended up with LTRs at either end. This then infects another cell, will express not just viral genes, but overexpresses oncogene, and leading to oncogenic development. There's a whole nother set of RNA tumor viruses caused by integration of these viruses into genomes. Usually that's because these LTRs are really good promoters. You remember there's also an LTR over here at the opposite end, right? So down at the 3' end. This is going to be transcribing whatever it happens to integrate next to. And this is the big problem that people had with some of these gene therapy using these lentiviral vectors, is that sure, you could express the genes you're interested in over here, but whatever gene you happen to land next to also is going to be being transcribed from this LTR. And that's known for a number of RNA tumor viruses, and so you've got both of these different kinds. Now, above and beyond these tumor viruses, now with genome sequence, what we know is that a huge amount of our genome is made up of particularly these endogenous proviruses. And the endogenous proviruses are basically a genome of a retrovirus. It's complete. It's got GAG, it's got Pol, it's got ENV, it's got LTRs. Somewhere between 8 and 10% of the human genome, and this is true for many vertebrate genomes, particularly the mammalian genomes, have 8 to 10% of these endogenous retroviruses, which are just inserted into the genome. So some point in our evolutionary history, these were inserted. Many of these have now some kind of mutations that have happened in them, so they're actually no longer active. So they can't actually form viruses. But in fact, one of the things that Harmit Malik, together with Michael Emmerman at the Hutch has done, has taken some of these endogenous retroviruses and actually made viruses out of them. Yeah?
Don't these eventually, over time, have the possibility of becoming engaged in regulation? Gene regulation elsewhere?
So I'm not sure I quite understand it. So is it possible that they could be involved in regulation of other genes? Well, it's not just possible, it's true. And so remember the example I gave you of syncytin, which is the gene that's critical for placental development. That's due to one of these. And what's happened here is it's that LTR right here, which is driving transcription with the little arrows right here of the genes right next to it. So it's that process, which seems to have led to the production of syncytin in this particular case. Yeah, so clearly some of these endogenous retroviruses are important for getting some kind of regulation. If you think about it, in 8% of the genome, if you think about it from an evolutionary point of view, this gives you a lot of potential genetic material that selection can act on. And so if anything, these can be really nice evolutionary agents. Above and beyond that are some of the retrotransposons, some of which have missing envelope genes, so retrotransposons that couldn't actually make an enveloped particle, but otherwise look okay. They've got GAG and PAL. So probably just deletions that have taken place in older retroviruses which were there. And then about almost half of our genome are these so-called line and sign elements, which replicate through reverse transcriptases, but they don't have GAG genes or envelope genes. And we can argue after class about whether these guys are really related to the viruses, which one came first. Was it these retrotransposons? Was it the viruses? I'm, of course, virocentric, so I think it came from the viruses first. But that's an open area of debate in terms of where these guys actually came from. But we clearly have lots of reverse transcriptases in our genomes and pretty much all other genomes. One thing just to mention here is that this particular kind is present in Saccharomyces cerevisiae. So it's true in lots and lots of different eukaryotes, not just the larger eukaryotes, as my yeast biologist friends like to remind me. So in the last 10, 15 minutes or so, I'd like to talk about HIV again. This is a subject, we could spend a whole class talking about. A couple of things, however, mostly in terms of not the disease, but more the differences between HIV and the other sort of classic retroviruses that we've talked about so far. Yes, there are huge numbers of deaths. 25 million is probably total at this point. Again, probably one of the major viral diseases, usually present at a relatively low load. If you look at the time course of a normal HIV infection, you get quite a lot of viral RNA up at the top here very early on. Then the virus RNA goes to a relatively low level. That seems to stay constant for literally years until something, and it's not at all clear what that something is, will lead to massive viral replication, and generally that's where you have your full-blown immunodeficiency. But this process of first infection and then really standard, apparently no real obvious phenotype. And so this can literally last for decades, and in some cases even longer than that. So generally relatively low load. The NEF mutants, if we have a chance, we'll talk about those right at the end. But in the cases, people, of course, are very interested in what that increase is all about. If you have mutants in the NEF gene of these retroviruses, often this line here just stays flat. So it's leaving very few of those. Cytotoxic T cells as well as the memory T cells, that question you had a little bit earlier. It seems that the response by the cytotoxic T cells may be important for that big increase at the end, but these are usually latent. They usually just hang out inside the cell, but you can still detect that a virus infection has taken place. And so if you think about an HIV test, what's the HIV test? It's basically looking for some of these proteins which are actually present inside the infected person. What are those proteins? Everything down to here is exactly the same as what we've talked about already. Matrix, capsid, nucleocapsid protease, reverse transcriptase integrase, SU and TM. The only thing which is different here is this VPR protein. But otherwise, everything else is exactly the same. The structure of these virions looks a little bit different. Doesn't have a usually icosahedral nucleocapsid structure in the middle. It's got this strange sort of cone-like structure that's present in the middle. And just literally in the last couple of years, we've gotten a really good handle on what that looks like. It's really kind of an icosahedron at one end with one T number and an icosahedron at the other end with a different T number that have somehow come together. So it's basically an icosahedral structure, but with these different symmetries at either end. Now, how that happens, lots of people are still trying to figure out what's going on with that. The 2 copies of the genome, tRNAs bound, etc. Otherwise, it's extremely similar. If you look at the setup of the genome, just like all the other retroviruses, it's GAG, POL, and ENV, but it's all these extra proteins that are present in the middle here, which are what makes HIV different from most of these other viruses. VIF, VPR, TAT, REV, VPU, and NEF. This NEF is those are the mutants that you have mutations in NEF. These seem to be these long-term persisters, people who can be infected with HIV, and as long as it doesn't have that NEF gene, it lasts for really long periods of time. The big thing that happens here is instead of just splicing to get envelope proteins, you also have splicing to generate all of these smaller proteins that you get in the 3' end of the genome. Again, all of our standard structural proteins, VPR, VIF, and VPU. Most of the time we're going to spend talking about TAT and REV in terms of the differences now with HIV. But first we need to talk about how HIV gets inside the cell. The main receptor is CD4, but there's also this co-receptor called CCR5. There are different kinds of HIV that have a slightly different co-receptor. CD4 is a really critical binding, extracellular binding protein that's present on lots of different immune system cells, and those people in immunology know way more about CD4 than me. But it's a really critical protein. If you have problems with CD4, your immune system has got all kinds of problems. On the other hand, CCR5, it's not quite clear what CCR5 is doing. And there are a number of people who are deficient in CCR5, and they seem to be perfectly normal, except that they can't get infected by HIV, which may, in fact, be a selective advantage. So that's you do seem to be able to survive with, again, an absence of CCR5. Just CD4 by itself, you can't get any kind of import that takes place. Unfortunately, a lot of the terminology in HIV, they give you the molecular weight of any of the individual proteins, which is really annoying. I'd much prefer they just call them ENV and TU and those proteins. GP120 is the surface protein that is present on the outside of HIV. This one is what's been a target for all kinds of potential anti-HIV drugs. Unfortunately, it undergoes large amounts of mutation, probably again because the reverse transcriptase has very low fidelity. So that low fidelity means that you've got all kinds of changes that happen in GP120, and so blocking the activity of GP120 is really hard. However, one thing that people have found actually very recently is that you can't get good antibodies to the end part of GP120, but it turns out that the stalk of GP120, remember where it's coming out of the membrane, that's where you're going to get that conformational change that happens when you get membrane fusion that takes place. Turns out that's really well conserved. And if you have mutations in those sequences, then the virus is not infectious, so it can't actually work. So what people are trying to do now, you may have heard about broadly neutralizing antibodies to HIV. Those are targeting the stem and sort of where GP41 and GP120 come together. And so this is a really active area of research right now. And so people are trying to develop and then hopefully be able to use these antibodies to treat HIV disease. MA is really important for getting the nucleocapsid inside the cell, but then there's one of these extra proteins that you don't find in the other retroviruses, which helps you get into the nucleus. So non-replicating cells can now get the viral integrase complex inside the nucleus because of the presence of this VPR protein. So VPR is important for getting inside the cell. That's actually present in your nucleocapsid because it has to be present in the nucleocapsid because you're not making anything until you've integrated your genome. Once you're in, there are a number of other proteins, VIF, VPU, TAT, and REV. I should put a big red box around TAT and REV because those are the only ones we're going to have a chance to talk about today. TAT is a protein which is serving as a transcriptional regulator, and that transcriptional regulator basically is an anti-termination process. And so what happens normally in the absence of the TAT protein is the cellular RNA polymerase will be transcribing the provirus DNA, which is present inside the genome, makes this secondary structure, also known as the TAR RNA, and then the polymerase dissociates. And this is potentially why lentiviruses are called lentiviruses, because they're slow. And unless you have the TAT protein around, which takes quite a long time to get more of that TAT protein, most of the time you get a short amount of transcription and everything falls off. On the other hand, if you do have enough of that TAT protein, together with a number of cellular proteins, particularly cyclin-dependent kinases and cyclins, which say, hey, we're going through the cell cycle. If we're going through the cell cycle, then we've also got a bunch of extra nucleic acids. We're actually a happy cell. We can make lots of proteins, which can then make a whole bunch of HIV virions, so it can get out. That process now leads to production of particularly full-length RNA. And that full-length RNA, that's what's going to get packaged into virions and exported outside of the cell. So you've made the full-length RNA due to the TAT protein. One of the problems that anytime you're making a messenger RNA inside the cell that gets spliced is usually you want to make sure that everything is spliced. And there's all kinds of regulatory mechanisms with HNRNPs and exon junction complexes, et cetera, which will make sure that non-spliced RNAs don't get outside of the nucleus. Well, if you're the virus genome, you want to make sure that your unspliced RNA does get out, because that's what needs to get packaged and moved along. How does that happen? It happens through the REV protein, which is one of these other extra proteins which is encoded in the HIV-1 genome. That then binds to, again, another sequence in the virus genome, and then gives you export now in the unspliced form. So this, of course, is in this region here, which otherwise would be spliced, where this REV response element is. So that will bind to your full-length RNA, and then allow that full-length RNA to be exported. And so it's a specific RNA-binding nuclear export protein, which will take this outside the cell. Whole bunch of really cool other important non-structural proteins, but we don't have time to talk about them today. So they won't be on the midterm.